A very good morning to Madam Principal, Honorable Speaker, Professor Shubhajit Bandyopadhyay, esteemed guests and participants. The Postgraduate Department of Chemistry, Lady Brabourne College, warmly welcomes you to their national webinar entitled Recent Advances in Chemical Sciences, Part 4. May I now request all participants to please mute their microphones and switch off their cameras. We would first have the inaugural address delivered by our respected principal, Professor Shivli Sarkar, who also happens to be the chief patron of this webinar. May I now request Madam Principal to please deliver her inaugural address. Over to you, ma'am.
Thank you, Papia. Respected Speaker, Professor Shubhajit Bandyopadhyay, respected organizers, that is the Postgraduate Department of Chemistry, headed by Dr. Papia Ganguly, Lady Brabourne College, respected participants and our beloved students, a very good morning and a warm welcome to all of you. It's a wonderful occasion when the Postgraduate Department of Chemistry has organized a one-day national webinar today, and the webinar titled Recent Advances in Chemical Sciences Part 4 is being started now. The topic is communicating with molecular machines using light. It's a pretty technical topic in chemical science, no doubt about it, and probably you all know by that time that science is not at all my cup of tea. So I will not waste much time by lingering my speech, and I'm not at all able to linger my speech either. I'm pretty certain that after Professor Bandyopadhyay's talk is over, there will be lots and lots of questions coming from the participants, and lots of their probable answers too are to be provided by our speaker today. And at the end of the webinar, I'm pretty certain everybody will feel more and more enriched and enlightened. Everybody will be able to know a lot more new things and a lot more new ways of explaining them. I wish today's national webinar a grand success. Thank you.
Today, we are honored to have with us our distinguished speaker, Professor Shubhajit Bandyopadhyay, who is Professor in Chemical Sciences at IISER, Kolkata, and presently the Associate Dean of Academic Affairs at IISER, Kolkata. Professor Shubhajit Bandyopadhyay, an acclaimed academician and eminent researcher, is going to speak on the topic communicating with molecular machines using light. The Industrial Revolution in the 19th century marked an epoch in the history of humankind, with large machines that could perform their functions. However, a similar comparison is often drawn in the early 21st century, which is marked by the advancement in miniaturization, opened up a new revolution in the idea of molecular machines. The international scientific community has recognized the importance of the area by awarding the Nobel Prize in Chemistry to Sauvage, Stoddart, and Feringa in 2016. These scientists have emphasized the challenges of controlling the movements of the molecular machines. In today's talk, Professor Bandyopadhyay will discuss his work in the area of communicating with the molecular devices using light and controlling their functions meaningfully. A brief intro to Professor Bandyopadhyay. Professor Shubhajit Bandyopadhyay, as I said, presently the Associate Dean of Academic Affairs and Professor in Chemical Sciences at IISER, Kolkata, obtained his MSc from IIT Kanpur and his doctoral degree in the area of molecular materials with Professor Reginald Michel of the University of Victoria, British Columbia, Canada. He did his postdoctoral work at Columbia University at the City of New York with Professor Ron Breslow, the father of biomimetic chemistry. Later, at the Department of Biochemistry at the University of Cambridge, United Kingdom, he worked as a postdoctoral research scientist. Apart from his interdisciplinary research in chemistry at IISER, Kolkata, Shubhajit Bandyopadhyay takes a keen interest in chemical education and pedagogy. He is active in the development of accessible technologies for education. And very importantly so, a smartphone-based app for students with visual disabilities developed in his lab is popularly used in over 30 countries all across the globe. Apart from teaching and research, his areas of interest include painting, studying history, literature, culinary activities, and even traveling. Professor Bandyopadhyay has been a recipient of very many prestigious awards and has received international acclaim and reckoning as a scientist of repute. He has received the Von Rudolph Award from the University of Victoria, Canada, has received the DART Research Stay Fellowship in 2012, has been selected for the INSA Australian Academy of Sciences Fellowship in 2013, has received the citation for the PhD Supervisor of Runners-Up at the Eli Lilly Top 10 Thesis Awards of Asia in 2014, has been recognized by the Royal Society of Chemistry as tomorrow's leaders in the field, invited publication in the New Talent issue of Organic and Biomolecular Chemistry in 2016. And last but not the least, Professor Bandyopadhyay has been selected thrice, not once, but thrice, as the best teacher by the student community of IISER, Kolkata. May I now request our esteemed speaker, Professor Shubhajit Bandyopadhyay, to please deliver his talk. Over to you, sir.
Thank you very much. Thank you very much, Professor Sarkar. And thank you very much, Professor Papiya Ganguly, for the invitation and also for your kind introduction. I felt that it was a bit too long. I don't really deserve that much, but thank you. And I also welcome...
I think you deserve it because you've been voted as the best teacher by the students. So definitely you deserve.
Thank you.
Congratulations once again.
Thank you. And I welcome all my colleagues, the professors, and today is a very special day for me because in the audience, I do see two of my very dear people. One is Professor Sanjeev Bagchi, who was the head of the department when I joined this new institute in 2008. And also my very old friend, Dr. Pradipto Ghosh, who is also associated with your college. And Pradipto is a very good friend of mine. He was my roommate at IIT Kanpur and an outstanding teacher, as I know. And I might not have passed some of the courses if he was not. So welcome Pradipto and Professor Bagchi. And I also welcome my dear students for this lecture. Now I'm quite informal. I will be talking about these molecular machines, how they are driven by light. I would switch to the PowerPoint slides now. And feel free to ask questions in the middle if you think it is necessary. I don't really mind at all, okay? All right, so let me get started. Now here you do see this QR code, which has all my details. So if you just scan this QR code with your smartphone, you can actually reach my website where you can find all about our research, our activities, and all the fun that we have in our lab. And let me move on. So I will start my talk with a story that might be of interest to not just chemists, to anyone in general. And the story is about two Buddhist monks. Okay, they're from two different sects, and in one sect there is a master, a guru, who is really famous for lots of tricks that he can do. So once these two guys met each other, one of them said, my master, my teacher is greater than your teacher. And then this other person says, why do you say so? He said, my teacher, my guru, actually could just wave his hand in the air and bring a glass of water or a bowl full of food. And he can wave his hand and there can be thunder and rain. He can wave his hand at the sun and the sun might disappear and the clouds might appear and so on. He just went gaga on and on about his own guru. And then he asked this other person, what can your guru do? What can your master do? And this other monk said, well, my guru eats when he is hungry and sleeps when he is sleepy. And now if you hear that, you might think that, oh, what's so great about this guru who can just sleep when he is sleepy and eat when he is hungry. But think of the level of complexity involved in any activity that we do. For example, the story that I have been narrating you, think of what is happening. First of all, these two monks, they had to walk up the mountains and they met each other. After they met each other, they saw each other and then they started the conversation. So think of the first level. Now, when you see someone, what are you doing? You are basically observing them because you have the light rays going through your eyes, hitting your retina, and inside your retina, you have a protein called rhodopsin, where you have a molecule called retinal, which undergoes 11-cis to all trans isomerization, which is a photochemical reaction. And because of that, the structure of that protein changes, which leads to a cascade of events, including a signal transduction through electrical impulse that goes through your nerves to the brain and gives you a perception of the vision, okay? And that is also coupled to all your receptors. If you see something scary, you move your hand, you move away. So that is also coupled to the other parts of your body, okay? Now, when you're listening to someone, for example, you're listening to me at this point of time, this sound wave is going and hitting your eardrum, and that is being translated to electrical energy. And then again, your brain is perceiving, and you understand what I am trying to communicate to you. And all these things are happening at the expense of the energy currency of our body, the ATP. Okay? And how is this ATP forming in the body? How is this ATP forming in the body? Let's look at the movie. This is a movie that I have actually taken from a web resource from Sigma Aldrich. Now think of this. This is basically a protein, an enzyme called the ATP synthase. Okay? In this ATP synthase enzyme, you basically have a proton pump, which is taking up the protons from the intermembrane space, pumping it to the ATP synthase, this rotor part, which is shown in orange. And as a result, there is some change happening here on the top. A molecule of ADP, adenosyl diphosphate, and an inorganic phosphate is coming and bind to this part of the molecule, as you can see in the cartoon. And then it is producing a molecule of ATP, your energy currency that you know from your grade 8. And as a result, the conformation of the protein is changing. The ATP that has been formed is going out of the system, and you are using that energy to do any activity, even walking around, listening to me, anything that you can think of. Now, I'll draw your attention to this part of the molecule, the protein. You can see very quietly in the animation a part is rotating. It's a rotor, right? And it's really a molecular rotor that is very much active in doing whatever we are trying to do by helping this whole cascade of event once again that is producing the ATP. Now, if you are thinking that this is the only example where we have a molecular machine inside our body, that's not true. We have plenty of molecular machines. I'll show you another one. Now, here we have the protein, kinesin, and dynein, both of them. And I'll show you these animations. These are, again, taken from, one is taken from the Hugenrad lab of the Utrecht University. The other one is by John Lever. Now, what you see here is this. Let's have a look at this. You see this. This is like a lame man with one leg walking along this rod. Okay. What is really happening is that this is the kinesin protein. This is the kinesin protein, and this is probably a vesicle that this protein is carrying. The thread-like thing is the protein. It is carrying the vesicle. And it can also carry certain other proteins, or it can carry large bodies like vesicles. And this rod that you are seeing here is a microtubule, right? On the right-hand side, you see this gentleman walking. This is also the protein dynein. And the dynein is also moving because of certain conformational change that is happening. I can walk you through the process, walk you through the walking process, where you can see the movement that is happening is because a molecule of ATP that was generated in the previous, if you have seen that in the last slide, is coming and binding here, and that is degrading to a molecule of ATP and an inorganic phosphate. And this conformational change is creating the movement of the protein, and with each movement, each step, a molecule of ATP is degraded. And as this is happening, the protein is hooking itself progressively to the microtubule, which is causing the movement. And you can see this huge load, whether it's a protein, whether it's a vesicle, or even a cellular organelle that is being carried away along the microtubule. And this is extremely, extremely important when you have the cellular division, which is right now happening inside my body and your body. Okay? So you understand that these molecular machines are very much operative inside our body right now as we are discussing its chemistry. All right. Now, these are the natural systems that are quite well known now. But what about the work of the chemists? Now, once again, look at this picture. This is a molecular car, okay? Molecular car, which has these wheels, and it is moving on a surface, a gold surface, basically. And this is a mimic of what happens in the natural system. A car, as Professor Ganguly was saying in her introduction, that we are going towards miniaturization. This is a miniature version of a car which is moving along this gold surface. Okay? Now, how does this work? Well, the principle for these molecular machines lie in many things. The machine can be light-driven. The machine can be chemical-driven. I'll give you examples of that in a few minutes. But let us see what happens in light-driven motions of molecules. The molecule that I'm showing you here is an azobenzene. Forgive me, there is a typo I see here. So this molecule, azobenzene, is something that was discovered well over 100 years ago. Okay, it's an old molecule. Now, if you shine ultraviolet light on this molecule, the trans form of the molecule, where you see all these phenyl rings far away from each other, they isomerize where the two phenyl rings are close to each other. So this is the animation of this. So in the cis form, the two units are close to each other, whereas in the trans form, the two phenyl units are far from each other. And this change happens with light. Ultraviolet light for the forward reaction from E to Z, and from Z to E, it is driven by visible light. Okay? And what is most important here is to observe that the structure of this molecule is changing. Okay, you can see this. Now, what Feringa has done is this molecule which operates with the same kind of principle, except for instead of the azobenzene, he has this molecule with a double bond here, a carbon-carbon double bond, which on exposure to light undergoes the cis-trans isomerization exactly like the azobenzene, but the structure of the molecule is different. So you can see this bold part going from the left-hand side to the right-hand side over here. And then once again, once you shine light of a different wavelength, it goes back to its original form. But note that this interconversion is not 100%. I'll go in a little bit more details of this. Now this is a more realistic picture of the molecule that was used in the car as the wheel, which can rotate. And here you see this molecule upon exposure to 365 nanometer light undergoes this isomerization. The trans molecule becomes cis. And those of you who are not chemists, just something that you can observe here in the picture is that this part of the molecule, which was on the left-hand side here, goes to the right-hand side, just flipping over, okay? And then thermally, at 60-degree temperature, this can rotate to a certain extent, and you have this conformation. It crosses this barrier, and then once again with 365 nanometer light, this can once again rotate like this, and at 60 degree, it forms a full cycle. So what you see here is this part, the black part of the molecule is static, whereas the red part of the molecule is rotating against this black part. So it's a molecular rotor once again, driven by light. This is Ben Feringa's work. And this is once again an animation. You'll see my talk is highly based on animation because we work with dynamic system. Molecules are not static. Our molecules actually move around with light. And that is why I have used quite a bit of animation, since we have undergraduate students who might not be familiar to all these processes, but I'm sure you're familiar with the cis-trans isomerization. And my talk is built up on that basic knowledge of cis-trans isomerization, okay? Now, if you are through with this animation and see how this bottom part is static and the top part is rotating nicely with light, this picture is clear to you. And you understand that we can attach a molecular car and we can drive the molecular car along the board surface by rotating the top part of the molecule, which are the wheels, okay? And these are the generations of these kind of molecular rotors that was basically developed by Ben Feringa. Some of the early ones starting from 977 to the later ones in 2006. Forgive me for the mistake. And then even there are some newer ones later on. Now, here is the picture of the molecular car that I have already shown you. Here is the chemical structure of the molecular car that I have not shown you, and you can see what I was talking about. This is the wheel which was rotating, okay, and driving the molecule. And this is another system which is built on a completely different principle. This is work by Fraser Stoddart, where you can see this molecule is protonated, okay? It has a charge here because of the protonation. Now you all know acid-base reaction, this nitrogen is protonated. If you add any base, this nitrogen would become deprotonated. Okay? It would lose the proton, one of it. And it would become neutral. So N plus would become neutral nitrogen. And as a result, the interaction that you see here with these oxygens would be lost. So you have two interactions here. These oxygen lone pairs would have some sort of a electrostatic interaction with the positive charge here, and there would also be some hydrogen bonding interaction. Once you get rid of the positive charge, this interaction will become weaker, and this part of the molecule that I have drawn with pink would move towards this end of the molecule where you have a stronger positive charge on these nitrogens, and you would have stronger interaction. So all you do is you take this molecule in solution and then add some base, and there is a change in the conformation. You can see this part of the molecule has switched and come towards this direction. You can go back by adding acid to this molecule, which would induce protonation in this part of the molecule. And the pink part would once again go back to its original state. Okay, so you can keep on adding acid and bases, which would take the molecule to this form or this form, this form or this form, back and forth, just by adding acid or base. This is once again a system where you can see the dynamics in a molecule just by adding some kind of a stimuli. Previously, you saw it was driven by light. In this case, you saw it is driven by a chemical, acid or bases. Okay? And in recognition of this work, in 2016, as Professor Ganguly was saying, that these three gentlemen, Bernard Feringa, Ben Feringa, Fraser Stoddart, and Pierre Sauvage, got the Nobel Prize in 2016. Now, these two gentlemen, the first two gentlemen you see in the picture, have a big impact in my own life. Fraser Stoddart was someone who actually gave me advice when I was a PhD student about choosing my postdoc. And that was really a great suggestion, and I'm really indebted to Fraser Stoddart for giving me suggestions. And that's that very important part of my life. And Ben Feringa is someone who actually cites our work on the topics that I will show you in a few minutes. Now, these are the Nobel laureates. Now I'll talk about someone who I think is probably going to be a future Nobel laureate, or if she doesn't become a Nobel laureate, it does not matter. You know who I'm talking about. This little girl, Greta Thunberg, she was in this 2019 United Nations Climate Action Summit in the city of New York. And there she made a remarkable statement. And what she said was this, I'll read it loud. You have stolen my dreams and my childhood with your empty words. We are in the beginning of a mass extinction, and all you can talk about is money and fairy tales of eternal economic growth. And she dares us and says, how dare you? And this is something, I'll be very honest, had shaken me at that point of time. That this little girl is of the damage that we are doing as various people. As chemists, I am doing it. As industrialists, there are many people who are doing it. As statesmen, there are people who are not doing anything to stop them. And of course, as statesmen, there are people who are actually promoting this damage. So I think this was a remarkable statement by a little girl, which we cannot ignore. And that brings me to the chemistry that we do. We actually try to take care of our environment with our own research. So before I go to the actual topic, I'll give you a very short glimpse of what we do in our lab. Of course, we work with this light-driven molecular machines. We work with sensing of toxic ions and molecules in the environment. We work on smartphone chemistry. Professor Ganguly, once again, just in her introduction, talked about this smartphone-based app that we have developed for people with visual disability. And other than that, there are many things that we do with smartphone chemistry. And I have also seen someone in the audience, Dr. Suchanda Chakraborty. She has done some remarkable work these days from your college. Fantastic. And of course, we also work on chemical education, developing tools for chemical education. And that's very relevant these days because I am giving my talk from my office room. You are listening to my talk from either from your home or from your office. And this means something, right? We are in an era of an unprecedented pandemic which has confined us to our small domains. We cannot go out. So it's very important that we continue our education and we develop new tools so that our students who are at home can access education through these. So we work on certain areas involving chemical education. Now, a little bit more on this work that we do. Why don't I show you pictures which are more interesting than just writing. So we work on monitoring water quality, and for that we develop fluorescent probes of various kinds. So we have basically fluorescent probes for mercury in water, we have fluorescent probes for this rocket fuel that is dumped in the environment, hydrazine, and this is another one where we can actually detect mercury in fish. You can see this picture over here by fluorescent output. And here are some of the publication records. This is a cover page of this journal where they showcased our work. And this is more recent work where with a single sensor, we can actually detect multiple toxic metal ions confidently in water samples. Okay? And these are some of the other works where again, we can detect some of the toxic analytes in water. For this work, we can use a smartphone and we can actually quantify this work. This is a picture of my student, Vishwajit Rai, who was heavily involved in this work. So we have inventory of number of fluorescent probes for monitoring water quality, and here are some of the publications, just to presentation. Other than that, we also work with monitoring the food quality, which is extremely important these days. I mean, anything that you can buy off the shelf these days in the market, starting from vegetables, fruits, anything, or even packaged meat, cheese, could be contaminated with many things, including pesticides. It could have bad bacteria in it, right? And for that, we have also developed certain chemicals which can monitor the food quality indirectly. So I'll briefly tell you what we have done in that area. So this is my student Atiur Rahman, who is now in Korea as a postdoc. So Atiur has developed this molecule in my lab where we have all these thiols, and in the presence of molecular oxygen in ambient light, this molecule polymerizes and makes this huge molecule, which is fluorescent. And I'll show you a picture of this. Okay. These are paneer samples, and this is a small chip that we have made with this fluorescent tag. If you don't have oxygen inside the package, it doesn't really glow that well. And this is the paneer sample. And we introduce oxygen deliberately in the sample, and you can see that this chip is glowing. Okay. And this is a zoomed in version of this. This is the same picture where you see in the absence of oxygen, you don't really see this chip glowing. If you have some oxygen, because of the formation of this large molecule, this system would glow. Okay, this gives an indication of whether there is a leakage of the plastic in the food sample, which is actually hosting the food sample. So this really costs a few paisa, and you can actually stick it to a food packaging unit, and then it will detect whether you have leakage of oxygen or not. And that is basically the indication of the food sample, whether it has gone bad or not. Okay, I can talk about this chemistry later on in the question answer session if you have any question regarding this. And this was recognized by DSIR, some organization by the government of India, which encouraged us to commercialize this. And my student Atiur and myself, we were in the newspaper, and this is a remarkable slide that we like to show that normally A.R. Rahman is always before Atiur Rahman, but in this case, the situation has been reversed. Atiur is first and then A.R. Rahman, and then there is someone who is very special. Normally the scientists are projected at the end, but this was a rare occasion where we were projected before all these other Bollywood stars. Okay. Now, keeping the jokes aside, let us move on to the actual topic of our research. And feel free to stop me anytime if you have questions. Okay. Now, I'll talk about some of the light-driven systems that were developed in our lab. I have kept representative examples. I have kept a molecular machine which catches metal ions or anions. This is for chemists. And a system where conductance could be switched. This is for physicists. And some enzyme mimics, which any biologist in the audience might like. So I don't know exactly who are there in the audience. I assume that there will be people of diverse interests, so I tried to keep it diverse. Now, I already showed you this slide where I told you that these molecules, as a benzene, can toggle back and forth between the cis form and the trans form. And again, there is this typo. I apologize for that. Now, this is Ben-Feringer's system that I have already showed you. Now, this is something that I have not shown you. This is the molecule that we make in our lab. This molecule is called a dihydropyridine system, a dimethyl dihydropyridine. Dimethyl because of these two methyl groups. And this is a pyridine system. This part is saturated, that's why it's dihydropyridine. Dimethyl dihydropyridine is this molecule, green in color. You shine light on it, visible light. It undergoes photoisomerization. So this bond that you see in the middle here breaks. See, in this form, there is no bond there. So it basically breaks there, and the green molecule becomes colorless. Okay? And this colorless molecule goes back to the green form when you shine this molecule with ultraviolet light or even with heat. Okay? Now, this molecule is the elder brother of the previous molecule that I have shown. The only difference is this molecule has a benzene ring fusion here. You see, here you didn't have this benzene ring. This elder brother has this benzene ring fusion. And this is a crystal structure of the molecule that definitely proves that I'm not really lying to you. And this is the photograph. This is the photograph of the system. This looks green. Sorry, this looks red. Once you fuse the benzene ring, because of the extended conjugation, the molecule becomes red from green. Okay. Now, if you shine visible light on this, this is once again an actual picture of this molecule, which loses its color. Why does it lose its color? I'm sure you understand that this molecule, being flat, aromatic molecule, has extended conjugation. Once you shine light on it, this molecule undergoes photoisomerization. This bond over here breaks. So this molecule becomes step-like. And the conjugation that you have, the pyridine system in the left-hand side molecule is lost here because this molecule is not planar. Okay? And that's why this is colorless. You can go back to its original form by using the ultraviolet light. Unlike Ben-Feringer's system, this conversion is quantitative, 100%. Okay? Now, if you understand this, the rest of the talk is going to be a piece of cake to you. Now, we thought, how can you use these kind of molecules and control metal binding, reversible metal binding? Now, the design principle of this molecule is as follows. We want one form of the molecule to bind to the metal ion very strongly. Once you shine light on it, I want this molecule to change its shape, which it does, and then this metal ion would be free. This is what we want. Now let us get into the actual chemistry. So this is precisely what we want. In one form, it should bind to metal ion strongly. In the other form, it just releases it. Okay, so this is a molecular tweezer that you can see. So it's very simple, probably the simplest of all molecular machines, a tweezer. Okay. Now, this is our molecule, this gives us the structural control for changing the shape of the molecule. And here we have the binder. You see the tweezer requires a binder, this part of the molecule. So the pyridine ring that you see here are the binders. The nitrogen donor centers can coordinate to the copper ion. Okay. Now, if you understand that, let me move on. We make this molecule, we have made this molecule, and then we arrange for this Swayamvar Sabha for this molecule and check who would this binder bind to. And it was found that indeed the copper ion shows this distinct change in the spectra when we basically scanned it against various metal ions. So we understand that copper definitely binds to this molecule, and some of the other metal ions that you see here might also bind to the molecule, but it doesn't give us a discernible change. Okay, so we proceeded with copper, and what we did was we bound the copper ion to this molecule. We have various evidences for that. I'm not showing you that because it's going to be quite boring, technical, but take my word for this. We have bound copper ion to this molecule, and then when it's bound, we use visible light. We shine visible light on this molecule, and it undergoes the photoisomerization. So this bond you see here breaks. There is a change in shape of the molecule. The distance between the two nitrogen centers increases. You can see it's quite close here. It is increasing over here, and the copper binding becomes loose, and the copper is released. Okay, and this is what we have done. We have measured the stability constant, which tells us how strongly this copper is bound to the molecule. In one of the forms, it's 81. This form, it's 81,000 mole inverse. And in this other form, it is only 7,200 mole inverse. Okay, so at least 10 times, roughly 10 times weaker binding in this form. So the tweezer is binding to the copper ion, the tweezer is releasing the copper ion, or at least if it's not releasing, it's not binding that strongly. Look at another aspect of this molecule, the same molecule. This is the molecular model based on the crystal structure. Here you can see the distance between the two nitrogen centers here is only 3.9 angstrom, 3.89 to be precise. When we shine light on it, the distance between this nitrogen center increases to 4.3. Now, what does this molecule remind you of? It looks like a human being, isn't it? And this man is basically moving back and forth as we shine this molecule with visible light and UV light. So we can toggle the distance between its legs by shining the light alternately. And we can say this molecule can run. However, I should warn you that it would not be very honest if I say that this molecule is just going forward and forward and forward like the animation that I have shown here. That's not entirely true because it is sort of random. So I could rather say that this molecule is thread in one moving. It's a particular man that has a long time ago, but his molecule, our molecules actually. To another topic. A molecular truck that can metal ion on demand. And once again, we use light for. So the molecule that we have chosen for delivery, benzoate ion, as you know, is a preservative that is used very often in the food industries. So when you are eating jelly or jam or various other food, they use benzoate as a preservative. It goes inside our body. So can we devise a delivery truck that can pick up the benzoate and then deliver it somewhere else? Okay. So this is just once again a simple cartoon. Now what we have done here is we have chosen a molecule, we have designed a molecule which has a very different principle of its action. Now, I don't know about the first year student, but I think from second year onwards, you people have studied this molecule called anthracene. One, if you fuse, it becomes naphthalene. If you fuse, it becomes anthracene. And 2 anthracene on either side of the molecule, and behold, molecule, there is an open space molecule, open cavity. Once we shine light on it, this molecule undergoes a zipper reaction. Means there is a chemical reaction between this unit in the next slide, and space becomes less. Okay, note this because the design principle of our molecule. Exactly what I was talking about the Diels-Alder reaction, pi plus 2 pi reaction, and it gives you a ring system from open. You have anthracene system which undergoes dimer. Well, it's not exactly the 2 units of the anthracene molecule, intramolecular dimerization, undergoes a 4 reaction in the presence of ultraviolet light, and from anthracene you basically put a bond between the 2 anthracene units, and we use this. And this is a theoretical possibility if you just take anthracene, but for our closed system like this, the number of isomers that you generate is going to be limited. Now, I have been telling you all these fictional stories. There would be people in the audience who would like to challenge us, and I will challenging us. Here are some of the evidences that we have in NMR. Since there are a lot of undergraduate students in the audience, I have chosen just a few exemplary slides from my usual research talk to show you that indeed this is not just fiction. There are very distinctly mapped evidences which shows that the binding, that the model that I'm showing you is right. And this is something that my friend Pradipta might like. This is a theoretical calculation which indeed shows the homolumo interactions between the 2 units, the benzoate and our host molecule. It's a favorable interaction that we can also see theoretically. In the closed form, the interaction is completely lost, and we can see that as well. Other spectroscopic evidences, I'm not really going into details of these unless you want me to. I'm just showing you that we can map how many units of the benzoate is bound to how many of the host units. It's 1 to 1. That is something we can see from Jobs plot. And I would very clearly acknowledge here that when I was a new faculty member, I didn't really know about Jobs plot. And I learned this from Professor Bakshi, who was almost like my teacher here. So thank you, sir. And other than that, we also have NMR evidences where we see that these molecules are binding and interacting very nicely. Okay, and this is the summary. So in this form, the molecule is binding to the benzoate, and once you shine ultraviolet light, this zipper reaction is taking place. It's like closing the zip. Okay, and there is not enough room for the benzoate here, not enough interaction to hold the benzoate here even. So the benzoate is leaving out. And then once again with 366 nanometer light, you go to this form, and then 254 nanometer, you go back. You can go back and forth n number of times. Now, we took a bigger challenge, and we asked ourselves, can we make a big molecule, big structure, and then break it down just by using light? This is an old work by my student Joydev, who is now in Germany. No, he has moved to Netherlands now from Germany. So basically what we are trying to address is that you make a big structure, you shine light on it, and it crumbles, it breaks down. Okay, and then once again you shine light on this broken down structure, and then can you go back to the original structure? So this is, you understand, this is something which is really a story of fiction in the macro world. You basically shine light on a house, it breaks down. You shine light of a different color, it again reforms itself. And you are asking this to happen repeatedly. Okay, so we cannot really make a big house and break it down with light, but can we make a big molecule and break it down and then again reform it? And here in this cartoon, I use this as the individual building blocks. I'll show you the chemical structure of this, okay? And these individual building blocks, once again, a simplified version. If you want to do this, if you want to achieve this, you basically require a big structural change in the molecule. A request for Professor Ganguly. Professor Ganguly, please warn me when my time is over, because I don't want this talk to be very lengthy and I don't want people to yawn, okay? It's very important that when I finish my talk, people should also have questions for me and they should not feel that, oh, why was it going on for too long, okay? They should not have that feeling. They should rather feel that, why did it finish? So warn me, please. Okay, now, for this structure, I have two big requirements. One is, if you want this molecule, the system to crumble, you need a large change in the shape. And you also need a glue to put these things together, a cement to put these things together. And that binding should not be too strong. Why is it? Well, you need to choose the correct binder, correct glue. If the binding is too strong, with a glue like this, then with light, it's so strong that it would not fall apart. And if it's too weak, it would not be a stable structure, right? So you need something in between. And this is my analogy. Now, the molecule that we have chosen, the unit that we have chosen is azobenzene. That is something that I have already shown you. It is like a molecular hinge, which goes back and forth by cis and trans isomerization. And this is the actual molecule that we have made in our lab. This molecule is long, 20 angstrom long molecule in the transform. And once you shine ultraviolet light on it, it becomes tiny because it puckers. Okay? And the distance between the nitrogen center becomes only 6 angstrom here. So a long molecule where the distance between the nitrogen centers are 20 angstrom becomes a tiny molecule just by shining light on it. And we have used metal ions once again to put these things together. And I'll show you a cartoon rather than showing the actual big molecules repeatedly because it might seem too boring. The glue was aluminum ion, okay? So here you see a nitrogen, and all these oxygens, they act as donor. Once you put aluminum ion with 3 positive charges, Al3+, there is a strong binding, a moderately strong binding rather, with the nitrogens and perhaps with these oxygens as well. And the second molecule of this sort would come in and bind on this side. Let me show you this. So what happens is that you put these aluminum ions, showed in red, and this is the molecule. You put the aluminum ion with this, and there is intermolecular interaction. So aluminum, azobenzene, aluminum, azobenzene, and so on. You basically form this long structure. And once you shine ultraviolet light on this, what happens is that this unit you see here, or all these units, would undergo cis-trans isomerization. The trans form would become the cis form, and the structure would crumble, and this coordination that you are seeing with many azobenzenes will become a one-to-one interaction. Okay, and we have wonderful evidences for that through various kind of spectroscopy and also with imaging. And you can again go back to the original form in the solution state. Okay. Now that brings me to an organic system, because sometimes organic chemists, often people ask me, are you organic chemist? Are you inorganic chemist? Why are you working with metal? Are you a physical chemist? Why are you working with all these spectroscopic methods? Now, this is a comment for my dear students. You should know one thing. Today, chemistry is at the crossroads where this clear line of demarcation between physical chemistry, organic chemistry, analytical chemistry, inorganic chemistry, all these demarcations are not that valid. I mean, we are comfortable going to other people's domain and working. I mean, I myself, being a student of IIT Kanpur, as my dear friend Pradipto is, we don't have any specialization. We try to specialize during our PhD period, but I was fortunate enough or unfortunate enough not to have any specialization. So I work in a very broad field, and I'm continuing in various fields, including organic chemistry, inorganic chemistry, partial spectroscopy, and analytical chemistry at the same time. Now, this system that I'm showing you is basically a supramolecular system where we can take this molecule, where instead of this benzene ring that you have already seen, is a naphthalene-fused naphthalene-fused dihydropyridine system called the naphthodihydropyridine system, in short, NDHP. You put a molecule that is electron deficient, pi deficient molecule. Once you put this yellow molecule along with this blue molecule, because of pi stacking interaction, a very well-known supramolecular interaction, this molecule will stack upon each other. And again, like this, okay? And once you shine visible light on it, this molecule changes its shape and the molecule would crumble. This is a publication that I had with my student Shiva Ramakrishna. Siva is here. He has already graduated. He is doing really well as a scientist in ISRO. And these are some of my older students. Here is Joydev, here is Moshami, this is Shumon, and this is me a couple of years ago. Now, let me take you to another story where we can make a molecular goalkeeper. This is something that I have taken from the web. It's not a great goalkeeper, but we have made a molecule which is actually a great goalkeeper. And for that, we have used a buckyball, the C60, and this is a big molecule. This work is done jointly with a collaborator who was in Australia before, and now he has come back to India in Goa University, Seshnath hosting. So this molecule once again undergoes C60 with UV light. In the you can see the two units, and once you put the C60 molecule then binds here and shine with visible light, the molecule or the technical goes out, and once again you have the free host molecule. So this is basically a molecular goalkeeper that you see. For the physicists, people who like physics, we have also made a material where you can control the conductance by light. And this work is by my student Monachura Shaha. He is going to graduate. He's at his final stage of his PhD. And these are some of the examples of photoswitchable molecules that were developed in our group, but also in some other groups across the world. This system is which was developed in Japan by IRA. It was also developed something that we have developed in our a molecule where you have a system done by my colleague Robinson with my PhD, Reginald Misch. And this molecule, once again, is a conductor with this dye that we work on. This was once again Michel's lab in Canada. Now, we use this azobenzene. And you know these azobenzene systems have certain drawbacks. These linear molecules, but the switching is not very fast. We have seen that there are certain molecules where you can tie two ends with a small bridge and in a much faster way. These are longer chains. Great. This molecule is a very short chain, not switch. So this is not really a great switch. However, in 2009, Harjes group, Rainer Harjes group in Germany, they developed the switch, which is really a one. We have developed this with a 9-member grain here in a fabulous switch. And this work was done by Monachura. The synthesis is for some organic chemist if you are interested in the synthesis, but let me move on. We have made this molecule. Here is the crystal structure, which tells us indeed we have made this molecule which can switch. And we have used this molecule to make a molecular wire with an insulator on the top. Okay? And this molecular wire requires a metal ion, and we have used silver ions here with an anion nitrate, which is basically bridging between the two silver ions. And here is the crystal structure of this molecule in the presence of silver ion. So it basically forms a complex first. All of you know this hard soft acid base principle. See, Ag plus ion being being soft, complexes with sulfur, not nitrogen. Okay, and you can see this is only one unit. The sulfur is bound to the silver ion, and then this is the anion, and so on. This is only one unit where you can see the silver ion, the nitrate, and two switches on either sides. This is a little bit bigger structure, and this is a full structure. You can see this is the metal part with the anion, and this is the organic part, which tells you that we do have a metal ion anion molecular wire, and this is the organic wire, which is the insulator. Now, this is very interesting, organic layer. Why? Because we have a switch, and as we switch, the interaction, the distance between the sulfur centers and the silver ion changes. As these units move away, it also pulls silver ions towards them, and this distance between the two silver centers increases, and this becomes a relatively bad conductor. And we have done, this is the size of the polymer as it grows. It's not really important. This is the SEM image, electron microscopy image of these materials that we have made. This is a control which shows that without the ion, you cannot really make it. In the presence of the ion, you can really make nice wires. And you take this and you basically make a device where you make the sandwich with the aluminum layer. This is our molecular layer. This is ITO layer, indium tin oxide layer. And here you basically connect this to a battery, and you can actually modulate the conductance. This is the IV graph just by shining light. And this is a cartoon that I have developed that you basically can open a switch and close a switch by using light, and accordingly the conductance of this material will change. Okay, I can actually stop my talk here if you are okay with it, or if you want to hear more, I'll show you one more small example just before finishing. So this is another similar system that we have made, where we can actually, this is the 2D polymer with metal ion, where once again we can actually switch the conductance between the two forms in a better way. Okay, this is not published. It's a hot result that I am showing you. Now, for something very small for the biologist, we have also made artificial enzymes. Now, after showing the very few first slides, you would definitely recognize our enzymes are molecular machines. So we have made several artificial enzymes which could be addressed by light. And the example that I'm going to show you is a carbonic anhydrase mimic that takes up carbon dioxide, and it can make carbonates, or it can take a carbonate ester and break it down. Okay, and this is the active site of the enzyme itself. You do see 3 histidines, which have these 3 imidazoles. You see the 5-membered rings with nitrogens, 3 of those, and then this is a zinc ion, and this is coordinated to a molecule of water. I'll show you the chemical structure of this in a second. So recognize 3 things. You need a zinc ion, you need these imidazoles, and you need a molecule of water. And this tapes that you see here is basically the other part of the enzyme. This work, once again, is done by my student, Mona Chua. So this is a molecule that we use where we have the imidazole units, the same unit as you see here. Now you can switch this molecule back and forth. This is the cis form of the molecule where we can bind to the zinc molecule in an acetate. just like this. And with this, we can actually do the catalysis. And this is the photograph where you can see that if you take the substrate and leave it with our artificial enzyme, it gives the product. This is monitored with spectroscopy. We have also found all these rate constants and stuff. I'm not going to details because the time is quite a bit already. And there are some evidences. We have also used a polymer. This is something that we have done with my friend Priyadarshide in the polymer lab. And we can show that this molecule works like a real enzyme. Okay? And we have also found all these rate constants and all these parameters that are used in enzyme catalysis. Okay, now that brings me to my concluding topic, not the concluding topic, concluding slide. Are we there yet? My own opinion is that this is what makes the most system that works inside our body, your body, my body. It's working at the. This is the level of sophistication, and our is where we are. This is something our children would make if they get a piece of paper. Okay, so we are far from this goal. However, this and even with these that we are making in Peringa's lab, my lab, are going to be very obsolete machines in the next 100 years. People would come up with more sophisticated machines, but ours is a good. That's all I would say. This is my team at the present. This is Sanjeev, who has done certain work. This is Shahid. Shahid's work I have not shown here. Monachura once again. This is Shahanawaz, Shilbhadra. This is Palash, Vishwajit, Atiur. And these are the funding agencies who have supported me. Unilever has supported me by giving us confidence. Let us not forget, supported by the Bharat mission, where certain apps for Bharat mission. And thank you very much for your attention that you have. Let me switch to there I am.
Thank you, Professor Bandyopadhyay.
I hope people are not yawning.
No, not at all, not at all. On the contrary, I mean, it had been a extremely interesting, illuminating, and a bit, something about which we have not much knowledge, the students and as well as us. It's absolutely enthralling. And thank you so very much for your time and just illuminating us with your lecture. Now the house is open for question answer. The first question which appears in the chat box is from Swati Thakur, postgraduate student of Lady Brabourne College. It is, can the molecular truck be used to deliver essential metal ions in case of deficiency like anemia, as it's very common in women?
Let me see the. Where is it? Have I crossed it already? Let me see.
Swati Thakur, if you open the chat box.
I will. Annesha Bhashyati Swagata, Dr. Ganguly, Dr. Basu.
No, you just.
Can the molecular truck be used to deliver essential metal ions in case of deficiency like anemia? Swati, you are right on. I mean, this is the long-term goal of this kind of work. I mean, what we have done definitely is a proof of principle. But think of two things. Number one, the molecule that we have made in our lab, we need to check whether this is biocompatible or not. Okay. And then the other thing is, can you actually use lights? The lights, the kind of lights that we are using inside our body? And the answer is a very clear no. No, we cannot. But that is the sort of things that ultimately would be our goal. So we are trying to develop switches which can operate with light, like infrared light, which can penetrate through our skin, through our tissues, and these switches would operate with this kind of lights, the infrared lights or near infrared lights. But this system that I have shown you in the slide would not work, very honestly.
Yeah, she has a second part of her question. Also, is drug delivery or toxic metal detection or their removal possible by merging these molecular machines with sensors?
Well, again, wonderful question. Thank you, Swati. Thank you very much. Yes. So whether these are possible at the present, I'll answer that first. Presently, the systems that we have theoretically can bind to a metal ion. Okay. What you really need is a delivery mechanism, as you have seen with this the kinesin protein, something that would be very targeted. It basically would like some toxic metal ion to be pulled up from a system, from a tissue, and take it to the kidneys from which it can be delivered. You can do it in a very untargeted way, just by putting, if you take some EDTA, it would complex with some of the metal ions that you have already in your body, and it would definitely take you the metal ion to the kidney. Now that might not be very, very targeted because it would go and bind to each and every metal ion that is possible. That's there in your body. But we can perhaps design molecules. We are not there yet. We can design certain molecules which can do this function. And that is once again the ultimate goal of this kind of research.
Thank you. I believe, Swati, you are satisfied with the answer. Now, the second question which has appeared is from Priya Poddar. She is our outgoing third-year student. Just like detecting food quality via fluorescent probe technique, can this technique be also put to use in detection of microorganisms like virus or bacteria in surface which are infected? I think this is based on corona, crisis.
Yeah, such a relevant question. Brilliant student.
Yeah.
Thank you for asking this question. Yes, it's very much possible. Indeed, it is very much possible. We have not really tried anything with coronavirus, but we do have molecules that can bind to the surface of the bacteria. I have not shown you those results here. That binds to the surface of the bacteria, and it can be mapped. Indeed. So, as you know, that there are two kinds of bacteria, where a gram-positive bacteria and gram-negative bacteria. So we can actually bind to some of the gram-positive bacteria, and we can actually visualize them. Indeed, this is very much possible.
So thank you. And another question from Srijita Roy, also a student. She has asked that Bernard Feringa mentioned the potential to use the molecular vehicle to inject into a bloodstream to find cancer cells. Is it possible in reality, and how far are we from this project?
Srijita, I think I partly answered your question when I answered the question by Priya. Now, this is still some trying, and to the best of my knowledge, that has not been achieved yet. But you see, this is the kind of research that is required to pursue. And perhaps people like you someday would actually help us achieving these kind of goals. Okay? So no, we're not there yet. There.
Okay, thank you. I am not being, yes, there is another question. Purbali Runu Das, she has asked that for making light-driven molecular machines artificially, what type of bonding connectivity in molecules, is needed?
Okay, Purbali, if you are on, would you like turning on your microphone? Because I would like to know exactly.
Please. Purbali, if you are there,
yeah, if you are around, turn on your microphone.
Please unmute yourself and please interact. Hello.
Sir,
Yeah.
hello.
Yes.
Sir, actually, I want to know what type of bonding connectivity. You said that azobenzene type. All the molecules have like azobenzene type connectivity. Is it really needed, or else other will do, like
Oh.
sulfur or any other type of molecules?
Okay, Purvali, I think in the first part of the talk, I showed certain azobenzenes. Other than that, there are various types of molecules that I have shown. One is this dihydropyridine kind of molecule where we broke the carbon-carbon bond by using light. There are other similar kind of molecules like the dithionylethene system that I have very quickly skipped through. There are n number of systems, and then there are some new ones which we have developed in our lab, which we have not published yet, which have a completely different connectivity. So you can have various kinds of bonds. The one that you mentioned, I heard you said sulfur. Yes, there could be molecules which would have sulfurs. I have shown you one. There could be molecules where you have nitrogens, and these sulfur, nitrogen, etc. would help these molecules to bind to other species. And these carbon-carbon bonds or nitrogen-nitrogen bonds that we have used in the photoswitches, they are basically operative intramolecularly to switch the molecule. Okay? So I hope I answered your question. If it's not clear, you can ask again.
No, sir, it's okay.
Thank you.
Okay, okay, thank you. I don't see any more questions in the chat box. If any of you present in the audience wants to ask any question, please unmute yourself and you may ask directly Professor Bandyopadhyay. Is there any more question? Hello?
Yes, please.
Yes, please. Is there any more question? Unmute. Swati, you are not audible. You are not audible. Then you write. You please put your question in the chat box. Nothing is audible. You just put it in the chat box. Meanwhile, I have a small question that is this whole idea based on the visual cycle phenomenon? I mean, the beginning of this EZ isomerization, has it started from the visual cycle phenomenon?
Well, yes and no. I mean, the visual cycle that we have is something that was naturally developed millions of years ago, right? Now, I can tell you some history. I mean, I personally am really interested in history, and I study quite a bit of history by myself, since I did not really have a chance to study it in the school that well. So during Alexander's period, in Alexander's army, sorry, something is happening. Okay. So Alexander's period, in Alexander's army, the generals used to use a bracelet, okay, which would have some chromic unit. Exactly what that was. It was a particular time of the day, the bracelet would switch its color. This general thought this was the right time to launch the battle. Okay.
Okay.
It's interesting because if you think, this perhaps meant that you had more ultraviolet light or a certain amount of ultraviolet light in the solar spectra would trigger this reaction in the bracelet when these people, they did not have the watch, but they realized this is the good time. It's like, in this Indian traditions of Shubha Muhurta. So this is basically, this was a photochromic system that was known. And then this azobenzene system was known before modern science, modern chemistry was very well known. This is like 100, 150-year-old chemistry. chemistry. So I'm basically teaching the old dog some new trick. And for many of the other people, but at the same time, we are also developing certain new switches that did not exist before. And I guess this is sort of related to what Swati about the topic. I mean, if you think involve color change, it's visually very appealing. And once again, I like to paint, attracted to colors. This is a very natural topic for someone who likes colors.
Chroma. I think I don't know, something has happened with the sound. You're not audible, but
okay.
No, but you haven't muted yourself, but I don't know why.
No, I'm not muted.
No, you haven't. You haven't. No, now it's clear. Now it's clear, I believe.
Okay.
Okay. Simply thank you so very much. Thank you so very much. I'm really, it was
there is a question, I cannot escape this. Is the chemical-based molecular motion mechanism by Stoddard more beneficial?
Beneficial in comparison to the photosensitized molecular motion mechanism?
By Olivia. Okay, Olivia, I don't think we can answer this in a very straightforward way. You know, the molecular switch that is chosen is based on the kind of stimuli that is available. For example, if you want to switch, if you want to have a molecular motion on the periphery where you have a lot of light, you can employ light. If you are doing something in certain cells where the H+ concentration is quite high, you can use Stoddard's motors. Okay, so the choice is completely dependent on the kind of places where you are trying to light. So I guess that answers your question. And then there is another question by Supriya. Supriya asked, in the earth, do we get plenty of UV light? How can we carry out this experiment? Wonderful. So we carry out this experiment in our laboratory where we use the artificial ultraviolet light sources or visible light sources. These are light sources that we use. We have various kinds of light sources, including lasers, and it's really something which is fun to do scientifically. Thank you for the questions. And let you wrap up, Dr. Basu.
Yes, thank you so very much. So if there are no more questions, then we may now conclude today's webinar. And once again, on behalf of Lady Brabourne College, I extend...
Hello,
I'm not audible.
Yes.
I have a silly question.
Yeah, sure.
I am Mustafizur Rahman. I have a silly question. How can you keep yourself very so simple? I saw you in Isaiah in very simplicity. How can you keep yourself so simple?
Well, it's kind of, you know, people say that beauty is in the eyes of the beholder. I mean, you are perhaps a very simple person and you see me in the same way. So thank you very much for your kind compliment.
Thank you.
Honestly, thank you so very much. And honestly, on behalf of Lady Brabourne College, I extend my sincerest thanks to Professor Bandyopadhyay for such a brilliant talk, and it presented in such a simple manner, honestly, in such a simple and lucid manner. I believe that all of us, and more so our students, they have been extremely benefited from this lecture. Thank you so very much for sparing your time. And we hope that in, I don't know when, near or distant future, we would be able to have you physically, not virtually, in our auditorium, in the auditorium of our college. So thank you. Thanks once again. And my thanks also goes to our Madam Principal, Professor Shivali Sarkar, for her constant support and encouragement in all our endeavors, not only this webinar, but in all our scientific and academic endeavors. And my special thanks goes to all the participants who have participated very enthusiastically. Thank you, my dear students, for being so attentive and putting up such nice questions. It has, made our effort a success, that you have been introduced to this excellent field, and the queries which have, cropped up in your mind will help you to initiate your research interests in this field. A big thanks to Professor Bandyopadhyay once again for this. The technical support team has also done a wonderful job. They also deserves a big thanks, and a big thank you to everyone for making this webinar a grand success. Thank you all.
Thank you very much. So